Genetics Matters, Episode 17, Genes that Regulate Other Genes. Last time on Genetics Matters, we were introduced to Jacques Monod, a French scientist who figured out that E. coli only made the enzyme for breaking down the milk sugar lactose when lactose was around, and when there was no better food to eat. Think about what that says. Not all genes are expressed all the time. You and I take that for granted today, but Monod and his co-workers were the first to figure it out. After determining that lactose induction of beta-galactosidase activity was a separate function than lactose acting as a substrate for the enzyme, Monod joined forces with two E. coli geneticists, Francois Jacob and Henri Lewolf. They first looked for mutants that could no longer use lactose as a sole carbon source. These mutations identified two genes, LAC-Z, which encodes beta-galactosidase, and LAC-Y, which encodes a membrane transporter called lactose permease. Loss of either gene's function prevents the use of lactose. These genes make sense in terms of using lactose, but what about induction? Well, it turns out that both LAC-Z and LAC-Y are only expressed when lactose is present and glucose is absent. How does E. coli know when lactose is around? How does glucose prevent lactose induction? Geneticists use mutants, but how to find induction mutants? It turned out that the lactose derivatives used earlier came in handy again. One of those derivatives, phenylgalactoside, or pGal for short, can be taken up by lactose permease and broken down by beta-galactosidase if and only if those two proteins have been made previously in response to an inducer. In other words, pGal is a substrate, but not an inducer. Wild-type E. coli cannot eat pGal as a sole carbon source, so Monod and co-workers looked for mutants that could. They found a handful of such mutants, and all the mutations mapped to one location, a gene that they called LAC-I, where I stands for induction. Mutations in LAC-I allow E. coli to grow on pGal. What does that mean? If removing LAC-I function allows growth on pGal, then the normal function of LAC-I gene must be to somehow prevent growth on pGal. But what does LAC-I act on? It turns out that in LAC-I minus mutants, both the LAC-Z gene and the LAC-Y gene are expressed all the time. Somehow, the normal function of the LAC-I gene is to keep the LAC-Z and LAC-Y genes off until lactose is around. The LAC-I gene was the first regulatory gene ever discovered. Yet how does LAC-I function, regulate LAC-Z and LAC-Y, and a third gene called LAC-A that I haven't mentioned before? What does induction of gene expression mean? The key experiment that answered these questions is called the Pajamo experiment because of the three scientists involved: Arthur Pardee, Francois Jacob, and Jacques Monod. The experiment involved moving good copies of both LAC-I and LAC-Z from a donor E. coli strain into a recipient E. coli strain that lacked working copies of LAC-I and LAC-Z. The experiment was done twice, once in the presence of lactose and once in its absence. At various times after transferring the good genes in, the scientists measured beta-galactosidase activity. In the presence of lactose, they found that beta-galactosidase activity went up over time, exactly what you would expect for lactose induction of the LAC-Z gene. In the absence of lactose, one would expect no LAC-Z gene expression, but that is not what they observed. Rather, beta-galactosidase increased early on after transfer of the LAC-I and LAC-Z genes, then leveled off. What was going on? If LAC-I was needed to get expression of the LAC-Z gene as a positive factor, then we would expect to see no induction because there is no lactose around to induce. On the other hand, if LAC-I was somehow keeping LAC-Z from being expressed, working as a negative factor, then when both genes come into the recipient cell, the LAC-Z gene can be expressed until enough LAC-I expression can shut it down. That is exactly what the Pajamo experiment found. It also means that lactose induction is the negative of a negative. It turns out that LAC-I encodes a protein that we call today LAC repressor. In the absence of lactose, it prevents transcription of the LAC-Z gene. When lactose gets into the cell, it binds to the LAC repressor and induces a shape change that removes the LAC repressor from the DNA, and the LAC-Z gene can now be transcribed. The LAC repressor not only controls expression of the LAC-Z gene, but also the expression of the LAC-Y and LAC-A genes. It can do this because these three genes lie right next to each other on the E. coli chromosome in the same direction. And they share one promoter. All three open reading frames end up on the same RNA. Monod, Jacob, and Lewolf called this arrangement of genes and coordinated regulation by the term operon. The era of studying gene regulation had begun. More next time on Genetics Matters.